The maize pangenome Grameen site homepage includes a search bar, links to specialized resources including our genome browser, the Plant Reactome portal and the EBI Plant Gene Expression Atlas. There are also links to our BLAST service, FTP site, YouTube channel, other Grameen sites, and MaizeGDB. The curated gene function link triggers a search for genes described in the scientific literature. Below this are links to some example views. Let's start by searching for the curated genes. The search results page is divided into a few components. On the left side, you can see the total number of genes and the current set of filters. Across the top is a visualization of each genome with at least one gene in the search results. Below this is the list of genes. Each has embedded views where you can explore different aspects of gene. Since we searched for curated genes, the Papers tab can be expanded to show papers describing the gene. Before we explore the other tabs, let's add another filter for genes in the jasmonic acid biosynthesis pathway. Click in the search bar and start typing. Matching search terms in various categories are shown, along with the number of associated genes and the taxonomic clade they belong to. When we choose jasmonic acid biosynthesis, 65 genes, a filter is added, so the search results now is 26 genes described in the literature and in the jasmonic acid biosynthesis pathway. If you wanted to change the logic to return the genes not described in the literature, click on the curated publication filter and negate it. You can see these genes have an Add Papers tab indicating they have not been curated yet. If you are aware of a paper describing a gene, you can submit one through the linked Google form and Grameen curators will integrate it into the next build of the search index. Let's delete this query and explore the other available views. The Germplasm tab lists genotyped accessions that harbor predicted loss of function alleles impacting the protein product. You can also click on the search button to find all genes with such alleles in a given accession. The variant image page lets you view all the variants within this gene region. There is a corresponding table view in which variants can be filtered in various ways. Let's filter by consequence type. The Sequences tab provides quick access to genomic, transcript, and peptide sequences. For genomic regions, you can expand the upstream and downstream flanking sequence up to 2 kb. The highlighted regions indicate UTRs and coding exons. You can click this icon to copy the displayed sequence to your clipboard. The Location tab provides a quick overview of the gene structure in which you can pan and zoom. Note these two transcripts use an alternative splice site, GCAG. There are also links to our Ensemble Genome browser instance and to MaizeGDB. The Expression tab has 3 ways to visualize gene expression. The default tab shows the EBI Expression Atlas reference study for the gene of interest and its paralogs. The All Studies tab shows expression profiles for only the gene of interest but across multiple baseline studies. Finally, the EFP Browser tab includes graphical views of several expression studies hosted by the Bioanalytic Resource at the University of Toronto. The Homology tab displays a gene family tree based on the protein sequence multiple alignment. You can click the gear icon to customize the way labels are displayed. Tree nodes can be expanded or collapsed. On the right side is a visualization of the multiple sequence alignment colored by Interpro domain. Click on a colored region to display details. You can resize and drag the area at the top to focus on specific parts of the MSA. Or switch display modes to explore the sequences directly. You can see this protein sequence is very highly conserved across maize varieties. The final display mode, Neighborhood Conservation, zooms out to show 10 flanking genes surrounding the members of the gene family. Each gene is assigned a color based on the gene family. Gray genes are from different families than appear in the neighborhood of the gene at the top of the display. If you click on a gene in the tree, you can click Focus on this gene to reorient the tree and reassign the gene family colors. By choosing an ortholog from an outgroup, you can see which gene families have remained in the neighborhood or moved away over the long term. This light orange gene is present in rice and sorghum, but may have moved away in maize. If you click on the rice gene ID, it will open in a new tab. Switching to the Neighborhood Conservation display mode and expanding the gene tree shows that this gene and a couple of other neighbors relocated to chromosome 9. Going back to LOX9, the Pathways tab displays an embedded pathway diagram and zooms into one of the reactions LOX9 is involved in. You can access a full-featured pathway toolkit at Plant Reactome by clicking on the links below the diagram. You can learn more about these tools by watching the tour. The Papers tab for LOX9 lists 2 publications. Finally, an XREFs tab provides a special link to search for the members of the maize pan gene set, as defined by MaizeGDB. Thank you for listening, and if you encounter issues, or have any questions or suggestions, please feel free to submit your feedback here.